Hey everyone, welcome to GeeklyEDU Biology. Today we're going to talk about the vitamin B12. In the 1920s, pernicious anemia was all the rage, well, for biologists. It was during this time period that treatment for this type of anemia was understood to be as simple as eating liver, especially raw liver. And in 1954, the chemical composition and structure of vitamin B12 was uncovered with X-ray diffraction crystallography. If you look at the chemical structure of B12, also known as cobalamin, you'll notice that it shares some structural similarities with heme and chlorophyll. As opposed to heme having iron and chlorophyll having magnesium within their respective nitrogen, oxygen, hydrogen, and carbon chains, B12 contains cobalt at its core, with phosphorus at the periphery, allowing for a few straggler elements. With the basic chemical structure understood, we need to uncover how our bodies absorb this miracle of chaotic atomic Legos and to understand what roles it plays within the mammalian physiology. While B12 can be found in certain fungi, the amount is absolutely dismal. The only realistic way to acquire the necessary amount, not accounting for vitamin supplements, is from animal products, most notably meats. Pernicious anemia is most commonly associated with those who do not consume animal products. Even with supplements, bodily chemical processes are required to turn it into something useful and biologically active. Speaking of vitamin supplements, the most common type of B12 that is manufactured to treat pernicious anemia and other B12 deficiency disorders is known as cyanocobalamin. The treatment can be applied orally, via muscular injection, or as a nasal spray. I would like to note that pernicious anemia isn't the only potential consequence of a deficiency in B12. If you want to learn more about cobalamin deficiency, check out this video by Armando. The link is in the description. Armando has been creating medical videos for more than a decade, and it's top-notch. Whether you're a medical student or a biology enthusiast, go subscribe to his channel and say hi. B12 is water-soluble, and if it is ingested in its free, non-protein-bound state, it will bind with a carrier protein called an R binder, or transcobalamin 1. Transcobalamin 1, a glycoprotein, also known as haptocorin or cobalophyllin, is secreted by the salivary glands in the oropharynx, or mucosal cells in the stomach. B12 will remain in this bound state until it reaches the small intestine. However, if B12 is ingested while protein-bound, it will bind with an R binder in the stomach. This process is dependent on pepsin. The process is known as proteolytic cleavage. Hydrochloric acid is necessary to convert the pepsinogen secreted by the stomach to pepsin. Pepsin then breaks down the protein containing B12, and the now free vitamin will bind with the R binder, transcobalamin 1. Regardless of how it's ingested, the duodenum, or the horseshoe-shaped portion of the small intestine, receives an R binder-bound B12, unless there are physiological digestive problems within the mammal that prevents this process. Once the second segment of the duodenum is reached, protease is released by the pancreas, which then breaks down the R binder, transcobalamin 1, containing the vitamin B12. At this stage, B12 will bind with intrinsic factor until it reaches the small intestine's ileum for absorption by the Q-BAM receptor. Intrinsic factor is a glycoprotein produced in the stomach by the parietal cells in humans or chief cells in rodents. Once there, B12 then binds with transcobalamin 2, where about half of it is delivered to the liver, and the rest will be delivered to the rest of the body that requires it. Interestingly, the liver's capacity to store B12 is enough to supply the body for potentially more than a year before malnutrition leading to pernicious anemia begins. There are several potential disorders that can affect B12 absorption, one of which is caused by an autoimmune response, wherein the body attacks its own parietal cells, resulting in intestinal cells replacing damaged or destroyed parietal cells. Without sufficient intrinsic factor, only a small percentage of B12 is bound and packaged for the ileum. However, for the rest of the passing B12, the ileum doesn't have the tools to process it, resulting in a B12 anemic disorder in the long term. To enter the bloodstream, B12 is transported by a protein within the ATP binding cassette transporter superfamily. The particular protein is known as ABCC1 or MRP1. MRP stands for multi-drug resistance proteins. MRP1 is a bit of a miracle protein that protects tissues from toxic molecules and secretes mediators that regulate inflammation. hormone secretion, and redox homeostasis. Now that we've discovered how it's processed and is transported through the body, let's discuss what it does. Cobalamin, B12, is required for the development, myelination, and function of the central nervous system. It is also required for healthy erythrocyte red blood cell formation and the synthesis of DNA. It functions as a cofactor for methionine synthase and L-methylmalonyl CoA mutase. Methionine synthase is a catalyst for the conversion of homocysteine to the amino acid methionine, or methionine, which is required for the healthy formation of S-adenosylmethionine, a universal methyl contributor for nearly 100 substrates. These include DNA, RNA, lipids, and proteins. L-methylmalonyl CoA mutase converts to succinyl CoA in the metabolism of propionate, a fatty acid with a short chain. There are two different forms of B12 once it becomes metabolically active: 5-deoxyadenosylcobalamin and methylcobalamin. They are known collectively as cobalamines. The other two forms, such as the form used in supplemental vitamins, cyanocobalamin, and the injectable form known as B12A, or hydroxycobalamin, only become biologically active after they are converted to one of the metabolically activated forms. To check out other awesome biology videos, check out our channel. Don't forget to like, share, subscribe, and hit the bell for notifications on future videos.